Okay, so just to remind you where we're at. So we're sort of halfway through this spectral library portion, right? So the spectral library was the second section out of 3. I cut some stuff out because I ran a little bit out of time yesterday, and I want to get through the spectral library stuff and get to the data analysis part because I think it's going to be more relevant for, particularly in the scope of this course. So here's where we stopped yesterday, but if you're following along, there's now a new set of slides for this second section, which Mina should have just uploaded. So if you want to follow along, take the new one on spectral libraries. So we'll go through that, and then we'll get to data analysis. Okay, so we talked about why yesterday. We mentioned why do we care about prior knowledge in targeted proteomics, so how can we utilize this? And now I'm going to just quickly mention how we actually generate these spectral libraries from a kind of high-level perspective, and then say a few words about what's available in terms of public library resources and whether or not it's a reasonable idea to use these. So essentially, when we're talking about spectral library generation, we're talking about some kind of version of this workflow, or at least this is how we've done it kind of classically. You'll hear later on, probably from Mike, I would say that there's possibilities to do this directly in the DIA data. I'll also mention that. Brendan mentioned DIA Umpire, so there are other options out there. But what I'm going to talk about first is just the way that we have done it mostly in the past, and we still do a lot of this, is to do DDA acquisition and kind of conventional database searching, and use this to generate a spectral library and use it as prior knowledge in the analysis of DIA data. So the workflow looks roughly like this. You acquire some DDA data. You do, optionally, usually you have to do some kind of data conversion and peak picking to generate peak lists to introduce into the search engine. You have to do a database search using whatever your favorite database search engine is today. Today in the tutorial, we're going to be looking at Comet searches. You want to do some kind of control, false discovery rate control of this database search. This is actually a fairly important step, such that whatever the spectral library that you generate is going to be reasonably high quality, and that you're not going to propagate errors at this stage into the DIA data analysis. You need to combine this database search results into some kind of spectral library, and this means essentially reducing the redundancy in the DDA database search results. And what that means is essentially that for any given peptide, sometimes you have more than one spectrum that matches that peptide. You have multiple instances, and you need to figure out a way to combine these. And then finally, we would filter this spectral library down to get what we call sometimes peptide query parameters, or the sort of coordinates for this peptide, or sometimes also referred to as assays. And this is what you've seen basically Brendan talking about in Skyline. On the left side of the Skyline document, what are the peptides that you're going to look for, what are the fragment lines for those peptides, and what are the kind of characteristics of those. And we generally, the question was how many fragment lines do we want to monitor, and we said roughly 5 or 6, right? And you have to choose which are the best fragment lines and so on. So you need a set of rules to filter these spectral libraries down to get the peptide query parameters. So this is the sort of workflow in a nutshell. This is the sort of complicated way of doing it, the way that we still kind of tend to do it in our lab, with a lot of, in terms of a pipeline, we use this TPP software. So some of you may be familiar with Peptide Prophet and tools like iProfit. We use Spectrast for the spectral library part. But I'm not going to go into really the deep details of this. This is referred to, I forget where, somewhere I have the reference for this protocol paper where we describe this in a lot of detail, but we don't really need to go into the low-level detail of this. It's just important, I think, more important for you to know what the steps are. And this is kind of the hard way, I would say. The easier way is using Skyline for the last portion of this workflow. In particular, the spectral library part. Skyline is using a tool from the Microsoft called BiblioSpec, which is doing the spectral library generation part. And this filtering has all these options are implemented in Skyline, and you can essentially plug in almost any search engine that's out there into Skyline. So if you use whatever your favorite search engine is, it will usually Skyline will usually understand these results. So the first choice you need to make here is what kind of samples you're going to analyze for the library generation. And there's several options here, but the easiest one really is just the samples that you're going to analyze in the DIA analysis, right? So you can simply take those samples, do DDA analysis of those, and that's the simplest place to start. Alternately, we found in the past that it makes sense because we see that DIA in most cases is somewhat more sensitive than DDA, and generally it's the case that in DDA it's not fast enough to cover everything that's in the sample. So sometimes it's interesting to take the samples of interest, or some representative samples of interest, or a pool of the samples that you have in your study, and to do some fractionation and do DDA analysis of these, and then you get a deeper representation of the peptides and proteins that are in your sample, and hopefully by doing this will be enough to exploit most of the information that's available from the DIA data. People do go other routes though, so many people have done synthetic peptides. So if you know what peptides you're interested in, you can synthesize these, and you can synthesize them in a kind of a crude way so that they don't need to be very pure. And this you can do pretty cheaply, so you can synthesize peptides on a solid phase format, in 96-well format for like $10 or $15 per peptide. So we've done quite a lot of this in the past. Or if you could get full-length recombinant proteins or synthetic proteins, for example, from in vitro transcription translation, is one way people like to do this, then this is actually also a very nice way of doing things, because in the synthetic peptide route you have to decide which peptides you're going to synthesize, and it's not always apparent in advance what the best peptides are going to be. So there are computational approaches to try and predict what the best peptides are going to be, but these have turned out not to work very well. So a safer bet is actually to if you can manage to do this, synthesize the full-length protein and just digest it and analyze it, and this tells you what are the best responding peptides and what are the best behaving peptides. But this requires obviously some effort to do this. And then the other one is basically public kind of organism scale libraries. So, and that's usually like some combination of all of these other four. So all the DDA data that's been acquired for a given organism, you can sort of mush it all together and make some kind of repository, and you can directly also use these, and these have kind of pros and cons, I would say. And essentially what we want to end up with is this spectral library, which we then turn into a set of peptide query parameters. This is the type of table I showed you already yesterday, the type of information that you need to know to extract data, extract quantitative data from DIA. And so I think this should be understood already. Brandon.
There's one more emerging one I think is that there are these assay portals where there's a varying level of like thoroughness in that CPTAC, the Clinical Proteomics Technology Assessment for Cancer, I guess they're now tumor something, but they've done a lot of vetting of certain assays. So you could go get the assays, and they've actually done reproducibility measurements, and they're highly vetted assays, and you can use those as spectral libraries.
Right. Good point. So I guess I would have put that in here, but there's definitely there's definitely different kinds of these, from sort of very discovery-oriented ones where people just dump data to very curated information. That's.
But they're not organism scale. I mean, the ones I'm talking about really are sort of very targeted assays.
Yeah. Okay, I agree. But they're public though. One of the words is right. And they are organized by organism, right? You wouldn't have them necessarily. So okay, now we're in semantics, but I'll change my slide. Okay, so if we kind of go down through this procedure, well, you'll see in the slides that I was going to present yesterday, there's a little bit more detailed information about these various steps, but I think this is probably mostly understood how to do a database search. I just want to point out one or two things that I think is important, right? So there's certainly the FDR control is very important, and we use this tool called Mayu, but there are many different ways to control the FDR using many different tools in DDA. Rudy brought up this point yesterday. I think it's worth kind of emphasizing again that you can have differences at the peptide spectrum match to peptide level to protein level. If you calculate the FDR at those different levels, then you can get different results, right? So this is a plot of the FDR at either of the false discovery rate, either at the protein level, the peptide level, or the PSM level. And what's on the x-axis here is well, this is iProfit score, but essentially you can think of this as a search engine score where the best stuff is one is best. And so if you take an FDR of 1% at the well, let's say you take an FDR of well, what's the best way to do this? An FDR of sort of 1% at the peptide at the PSM level. This would result in an FDR of like 2% at the peptide level, but actually something like 8% at the protein level. So as Rudy mentioned yesterday, for this reason that the PSMs, the true ones, tend to distribute themselves in a non-random way in proteins. So here, each one of these little circles is a PSM, and the hexagons are the proteins, right? So the green ones, the correct ones, tend to build up on certain proteins, but the incorrect ones tend to distribute themselves across many different proteins, and that's why you have this disparity between false discovery rates at different levels. So it's worth calculating the false discovery rate at these different levels in order to make sure that the spectral library that you come out with in the end is controlled at the quality that you want. So the question is, why do we need to control the quality of the spectrum library so well? Well, let's consider what would happen if you had errors in your spectral library. So here's like a DDA spectrum, and let's just imagine for whatever reason that the search engine assigned the wrong peptide sequence to the spectrum because the spectrum wasn't great quality or whatever, but for stochastic reasons it got some kind of a decent score, and we got the wrong peptide sequence assigned. So what would happen when you then use that spectrum to generate peptide query parameters and try to extract that peptide from DIA data? Well, what's going to happen is that if this spectrum is just some other peptide which you wrongly assigned the sequence to, well, then you're going to recover that same peptide in the DIA data, right? You're going to get a very coherent match between this set of information in the spectrum library, the spectral library, and the set of information that you extract from the DDA data. So the error that you made is going to be propagated from the spectral library into the SWATH, the DIA data analysis. It could also be the case that something else would happen, like this peptide sequence was wrongly assigned for a different reason, like maybe it was a chimeric spectrum, so you co-isolated two peptides, and just because you got a little bit unlucky, the combination of these different fragment ions matched with a reasonable score to some other peptide, so you still have a wrong peptide sequence assigned in the DDA data, but it's for a slightly different reason. And then when you extract this from DIA, well, it's possible, or probably I would say it's even likely, that you will get two different signals in the DIA data, and this should be resolved, right? So this type of signal, if the scoring system thinks all these fragment ions belong together in one peptide and you get two different chromatographic signals, well, that's not going to score well. So in this case, the error would be resolved and this would drop out. But the problem is we don't really know what's the prevalence of different kinds of errors in the DDA data. So it's sort of reasonable to be conservative and try to control the quality of these spectral libraries as well as you can, such that you don't propagate these kinds of errors into DIA. Okay, so I want to say something about iRT. So Brendan mentioned yesterday retention time, and that this can be, we said that this can be a powerful score in determining whether the signal that you're looking at corresponds to the peptide that you're looking for. The issue is, of course, that as you guys all probably know, that the chromatographic conditions can change from day to day, or week to week, or instrument to instrument, or chromatography setup, column, and mobile phase, and so on. And the question is whether we can. So these retention times in absolute terms. This, if it's 11 minutes one day, well, it might be like 15 minutes 2 weeks later. And how do you use that information? So we need some way to kind of standardize this retention time scale and make the information transferable between analyses. And one way people have done this in the past is to include some standards which have known chromatographic behaviors. So this is the thing that we're interested in, but we're now going to spike in two peptides into this sample, which are standard A and standard B, and these are kind of well characterized in terms of their chromatographic behavior. And instead of saying that this peptide comes out at 11 minutes, what instead we're going to say is that essentially the index or the distance from between A and B, where our analyte is like 60%. So if we call this 0 and we call this 100, well, then the analyte that we're interested in is 60, right? And these red numbers are on some arbitrary scale. It doesn't matter what this scale is. It's just some numbers to say this is kind of like 60% of the way between A and B. And then if the next time you do this analysis, you include this standard A and standard B, well, that allows you to predict where this yellow guy should be, because you now know that it should just be in between A and B and roughly a little bit closer to B, essentially. So this is the concept of a retention time index, and this is a number that we use to store the retention time of an analyte relative to some other set of standards. And so this is the set of peptides that we typically use. This is a set of peptides that you can buy from Biognosys, but there are now several other commercial kits available, and you can you can also use actually any set of peptides that you're pretty sure are going to be in your sample, and you'll see in Skyline that there are several different sets of peptides that are supported. But essentially, this is the one that we use in our lab, and it's just a set of 11 peptides which are distributed nicely across the gradient with sort of relatively even spacing. And by spiking this into every sample that we analyze, both on the library generation side and on the DIA side, it allows us to make projections from one data set to the other and make predictions about, yeah.
Would you recommend using endogenous peptides to do peptides?
Yeah, well, so this actually works pretty well. So, yeah, I mean, this came up for us at one point when there was a postdoc visiting us from Jenny Van Eyckslab, Sarah Parker, and she came with a bunch of data that didn't have any peptides spiked into it. So we routinely do use this set of iRT peptides, which you spike into all the samples, but she came with data that didn't have it, and so we had to adapt our kind of software pipelines to try and make this work without those peptides. And what she did was I guess you're referring to this paper, this CIRT paper, where she looked for sets of peptides that come up all the time in most kinds of samples. And so basically this works, as far as I can tell. I haven't really come across a sample where it doesn't work properly, including like phospho-enriched samples or pull down samples or samples from like different species. You still find actually most of these set of peptides. They're just everywhere because they come from like trypsin or keratin or some filamentous proteins that are just everywhere. And so this has actually never failed in my hands. So I think it's reasonably safe to do. With that said, we still spike in these peptides because it's just a convenience. It's a useful way to do it, and we know they're always going to be there. And we actually sometimes use this set of peptides for quality evaluation as well, because we know how they should perform, right? And it's pretty fast to kind of look at this set of peptides and see what their signal is and see whether something's wrong with our instrument as well. So they kind of serve another purpose too. But I don't think there's anything wrong with using these CIRT peptides. But you know, I can't guarantee that it's going to work every time. This is basically guaranteed to work every time. Okay, and so the scale here is just again some arbitrary number. This is a unitless number where they set the second peptide to 0 and the last peptide to 100, and you can say, oh, why is this minus? Well, the reason is that this first peptide is slightly unstable in chromatographic terms, and so they decided it was better to have 0 as a second peptide, but that's just a kind of a legacy reason. And so what you do is, when you're building the spectral library, you spike this set of peptides into all the samples where you want to do the where you want to generate the library, and you extract the ion chromatograms for this set of peptides, and you figure out what retention time that they're at, and then now you make a regression of the observed retention time here on the y-axis versus this iRT value, which is predefined basically in the kit that they give you, or in the CIRT paper, or in this drop-down table in Skyline. And essentially, what you're going to do is project. And then, so this is the regression that you have now, and then when you have actual peptides that you're interested in, so here's peptides A, B, and C that we're interested in, and we're going to look at what the retention time of these peptides are in absolute terms in any given run in the library generation, and we're going to project it onto this regression and project it onto the iRT axis. And now, so we have these values for these peptides A, B, and C. We have the so-called iRT values for these peptides, and that's what we actually store in this peptide query parameters or in the spectral library, right? So we store this information in this normalized iRT space, and that makes it now portable for when we want to go and apply it to some other data. And so what do we do when we actually have DIA data? Well, so let's imagine now this is now the DIA data, and notice that here I had a 70-minute gradient, but actually you can use a completely different gradient. This sort of compression expansion of the gradient doesn't really matter. But what you want to do when you want to look for these peptides of interest is to say, okay, now in the DIA data, I've also spiked in these iRT standards, and I can again make this regression, right? So these are the values for my iRT peptide standards in absolute terms in my DIA data. I'm going to make this regression again with these iRT values that I know. And now I'm going to, for these peptides A, B, and C that I'm interested in, I'm going to go from the iRT values and I'm going to project it back through the regression to make a prediction about what the retention time of these peptides A, B, and C should be in the retention time space of the run, this single DIA run that I'm interested in. So now we're making a prediction. So notice that in the library, these peptides were at like 20-something minutes, 50-something minutes, and 60 minutes. And now we're predicting that in the DIA data, in this shorter gradient, they're going to appear at sort of 12 minutes, 25, and 30 minutes, right? So this is how we are making this retention time information portable by standardizing it with respect to some set of known marker peptides. And this allows us to then, in the DIA data, to say, oh, I'm only going to look at this retention time for this particular peptide, and here I'm going to look at this particular retention time for this particular peptide, and it's also going to allow us to score whether this peak appears at the expected retention time, and that's one of the things that we're going to score in terms of when we want to say whether this is the peptide that we're actually looking for or not. So this is this delta iRT that Brendan mentioned yesterday. And then the last step here would be just to take this spectral library and make some choices about what fragmentines that we're going to monitor. So we said we usually use 5 or 6 fragmentines per precursor in this charge states in a particular mass range. Usually we can use B and Y ions, assuming that it's label free. If it's got heavy labeling, then you might have to kick out the B ions if you have a C-terminal labeling, like arginine and lysine labeling, for example. This is a bit of a detail. You can, in some cases, use neutral losses, but this is done with some care, as Brendan mentioned. We usually are excluding fragmentines which are in the precursor isolation window because these tend to be rather noisy. And basically, for our software pipeline, you need to add decoys. Skyline will do this internally. But essentially, this is now where we are, right? This is the table that we're looking for, and this is the table that we now need, and this table can be imported into any one of these different softwares. We're going to use Skyline today, and this we usually store in some kind of table format like TSV, although there is an XML format which is supported by the Hoopo PSI, although as far as I can tell, nobody really cares about it, but we also support this in Open SWATH. And so this is the goal, this is where you want to get to in terms of this library generation. And all that stuff that I just described now, you're going to do in the tutorial, but it's going to take you like 2 minutes to do in Skyline. We're going to start from pre-searched DDA files that we've searched from Comet, and you're just going to build a spectral library from that and make these peptide query parameters, and that's all going to be done inside of Skyline. So briefly, just to mention the idea that we could also make spectral libraries directly from the DIA data, from the same DIA data. So this is something Brendan mentioned earlier in terms of DIA Umpire, and something also the McCoss Lab is doing in terms of this Pecan tool, which I think Mike will probably talk about later on. So let's remember again the problem. So here's a SWATH or a DIA spectrum. And remember I said, okay, we know that there's a particular peptide in here, and here's these fragmentines that we know should be from this peptide, but they're like these low, they're not the most dominant fragmentines in the spectrum. So if we try and search this thing with like Mascot or Comet or something like this, well, it's not going to work. But is there any way we can massage the data well, I shouldn't say massage. Is there any way we can process the data in a way that will allow it to be interpreted by classical DDA searching approaches? And the answer is hopefully yes. So here, for example, is a kind of a toy example where we have these 3 peptides. They all come in, they're all falling in this one precursor isolation window, right? So we fragment them all together. We get this complicated mixture spectrum, and this is sort of helpfully colored. The fragments are colored from which precursor they came from, but obviously you don't really know this in a real situation. But there might be a way to reconstruct this information by using chromatographic elution information. So what we would do here, and this is what has been done by Waters for a long time, and now also by DA Empire, is to say we're going to extract ion chromatograms for all of the features that we see here. We're going to extract ion chromatograms for all the features that we see here, and we're going to see if they co-elute, right? So what happens is that basically the red ones are going to co-elute with the red fragment ions, and we can, using that correlation, we can assign these red fragment ions to this red precursor. And so this is essentially a deconvolution step. We're trying to take the fragment ions here and match them together to this precursor. And now once you've done this deconvolution, now what you have is kind of what we call pseudospectra. So we have this yellow fragment ions, which we think belong to this yellow precursor. And now that's something you can search with a regular DDA search engine like Comet or Mascot or whatever. And this will return the type of information that you're probably used to seeing from DDA results. And then we can use that to do what I just described previously and build a spectral library and use that spectral library to interrogate the DIA data. But this is all happening, this building of the spectral library, this deconvolution, the building of the spectral library, and then the extraction from the DIA data is all happening on one DIA file, right? Or one set of DIA files. And so this is actually a workflow that we've used quite a bit in our lab. And it works pretty well. We still, at this stage, don't see it as sensitive as doing like a deep DDA spectral library based on fractionation. So we still see some benefit to doing that, although I think as the DIA data gets better and more high quality and higher selectivity and kind of smaller isolation windows, I think that gap will probably close to some degree. Brandon.
It does rely on recognizing precursors.
Yes.
So you need to recognize a precursor peak, and so like that example I showed yesterday where the two precursors were not necessarily resolved from each other, like where we got a shoulder in the middle section, that kind of thing will mess up this type of detection, right? It's not as like for DDA, you're dependent on the instrument recognizing that there's a peak to isolate. But for DIA, for this kind of this is not dependent on the instrument. It can be done by a computer later, but that computer still needs to recognize that, oh, that thing is actually a separate precursor and not just the shoulder. So it will foil it, and yeah, in our lab we are developing tools that will look only at the fragment ion spectra, and we are finding things where we can't see a precursor.
I agree with all that. But still, to a first approximation, it works. It works to a sort of 80%. But yeah. Oh, sorry. Yeah.
We improve it by the high mobility side.
Oh yeah. Yes, that will definitely solve like some of the problems that Brendan mentioned, where you have two things sitting on top of each other. The ion mobility could solve that. It's still the case, though, that sometimes we just see better sensitivity for the MS2 data than we do for the MS1 data, and we just don't see a precursor there. It is actually possible to still search probably the MS2 data without knowing the precursor information, although that's going to also be problematic. So, yeah, so the ion mobility, this is what Waters have done for years. So are you talking about Waters or the Brooker? Yep. Yes, I think that will help too. But I think it's early days with Brooker. But I'm also excited about what will happen with this Brooker instrument. That's a whole other conversation. Okay, so what I've kind of described here is, well, our kind of most routine workflow and what we use in the lab all the time is we start from some kind of prior knowledge in the form of a spectral library that we've generated using DDA analysis, and we use that spectral library to generate peptide queries. So what are the coordinates for the peptides that we want to look for, and then we use that to analyze the DIA data. What I've just said is that it's possible to maybe go from the DIA data to the spectral library, right? This is like using the DIA Empire approach, and what Brendan talked about and what probably Mike will mention later is to directly make queries of the DIA data using kind of in silico generated coordinates for the peptides of interest. So skip the prior empirical knowledge, but just say we predict what these peptides are going to look like by just looking at the sequence of the proteins and the peptides, and just go looking for those in the DIA data. And I think that these two methods are going to benefit from, as the DIA data gets better, these two methods are going to benefit from having higher quality DIA data. And actually, as Brendan mentioned, we saw that already in this LFQ bench study where they compared like 32 window data with fixed windows to 64 variable window data. And for example, DIA Umpire benefited disproportionately with respect to the other tools in terms of how much of a boost it got from going to this higher selectivity data. And I think also I think that will be true for, I think Mike has mentioned this is essentially true for Pecan as well. When you go down to lower precursor isolation windows, it makes this kind of Pecan approach more viable. Okay, so I'll make a few comments about publicly available spectral library resources. And so, well, yeah, at some point we noticed we were doing, SWATH project number 1. We had to generate assay library number 1 in order to do SWATH analysis number 1, and then we had project 2 and 3. And then we thought, well, why don't we just aggregate all these libraries together, because we're making different spectral libraries for each sample of interest. So let's try and aggregate them and see what happens. And that's essentially what we did. So this was an early effort to do this, where we took all of the human samples in the lab that had been analyzed by DDA, which was at the time around 330 runs, using all sorts of different cell lines, HEC293, using pull downs, SEC fractionation, different other cell lines, tissues, blood plasma, whatever, basically whatever we had, and we combined them into a single spectral library. And so that's what's shown in this kind of blue circle here. It represents about 10,000 proteins, and we call this combined assay library, or sometimes pan-human library, or whatever, but it's basically our big human library. And so if you compare that to what's in SwissProt, there's about 20,000 proteins in SwissProt, and we cover about half of those. And then what's green here is the proteins that have protein level evidence in SwissProt. So we see there's a strong overlap with what we detect and what other people have detected in the past. So that's kind of gives us some confidence. And this is basically how many proteins and peptides and precursors and transitions are in the spectral library, and you can potentially download and use that if you want to. Where you can download it from is this portal called SWATH Atlas, which is maintained by Rob Moritz's group at ISB. And basically, this is a whole bunch of, it's basically where we put these spectral libraries. It's not particularly fancy place. It's basically a dump for the data. But here you see this 10,000 protein library. We also have HLA peptides and a bunch of different libraries in other organisms. We've done a fair amount of work in M. tuberculosis, and other people have done other work in other bacteria. And so this is basically all downloadable in either the spectral library format or else these kind of table formats where you can directly plug them into software for doing DIA analysis. So this is not particularly sophisticated portal, but the data is there if you want to try.
Do these databases have the iRTs spiked into them?
Yeah, I think they should basically all, yeah. Or if they don't, so they should have the iRT values for all of the peptides, but they might have been generated using like this custom iRT set of peptides or other sets of iRT peptides. But I think most of them should have the Biognosis 11 peptides spiked in, as far at least the stuff from our lab. But there is some stuff from other people's labs too. Other resources, so I mentioned people have been trying to do this with large-scale synthesis of synthetic peptides. So our lab used to do a fair amount of this along with ISB in Seattle, and we have a few different species here, human, tuberculosis, and yeast. This is mostly data collected on QTOF. I think it was an Agilent QTOF, actually. And this is an interesting resource, but again, this is kind of to be used with some degree of caution, because the peptides, they don't synthesize all peptides for all proteins. They synthesized a handful of peptides for all proteins. And that means that they had to choose which peptides they were. So this was chosen to some degree on empirical evidence which peptides had previously been observed. But when that wasn't available, they had to use an algorithm to try and predict what the best peptides would be. And as I said, it turns out that these don't really work particularly effectively. So the set of peptides that were synthesized are probably non-optimal. So I would consider this as kind of a supplementary resource. I wouldn't necessarily use it as a kind of primary resource. Bernhard Kuster's lab in Munich has now gone and done the same thing, except on a much larger scale. So here there was like 150,000 peptides for human. He's now up to 300,000 peptides, tryptic peptides, which he synthesized for human. But they're also, and this is the part that's published, but they're also synthesizing all sorts of variants, like genomic SNPs variants, post-translationally modified peptides, non-tryptic peptides, other peptides like MHC peptides and this kind of thing. And most of this stuff is not published yet, but eventually they're going to have like, more, I don't know how many this adds up to, but more than 1 million peptides synthesized, and they host it on this Proteome Tools resource. This I don't think has iRT inside, but you could probably figure out that. If you worked hard, you could do some transformations by having peptides that you know the iRTs for and doing recalibration, but I don't think it's there in a convenient way. This has mostly been collected on Orbitrap and IonTrap using both HCD and resonance CID modes, and with also a lot of different activation energies. So one question that comes up, I should say also, Brendan mentioned more curated repository. So Panorama, the one that's linked to Skyline, is also a more curated repository. He mentioned the CPTAC portal, which has also very curated information. I also mentioned this Passport resource from the McCoss lab. And this one is interesting because, as I mentioned at the start, they're using full-length proteins from in vitro, like cell-free transcription translation systems. And that means you don't have to make any decisions about what peptides you're going to use, because you just digest a protein and see which peptides are the best. So that's actually fairly attractive. There's not a whole lot of proteins on this resource yet, but I think it's a neat idea in terms of how to produce good information. One question people ask a lot is, can you move between instrument types? And the answer is probably yes, but with some care. And this is not like a black and white thing, like yes or no question. It's that some instruments will be more or less like each other. So what I'm showing here is a comparison of a spectrum from an Orbitrap. So the top one is QTOF, and the bottom one is Orbitrap using HCD fragmentation. And what you can see is basically they're almost exactly the same spectra, except for with the exception of a couple of B ions up here. And this is now, if you look at this over comparing Orbitrap and QTOF, when you have the kind of collision energies properly aligned and so on, it's actually very highly correlated in terms of their relative fragment ion intensities. So the answer is you can do this if you do it carefully. And generally the kind of rules of thumb would be like things that use beam type CID, like triple quads or QTOFs or Orbitraps in HCD mode, will produce very similar spectra in terms of their relative fragment ion intensities. If you then go to resonant excitation like IonTrap CID in IonTraps or IonTrap Orbitraps, well, this is going to be more different, and there's some evidence that you can, for example, use the Y-ion spectra, the Y-ion intensities from this type of spectra. And then if you go to more exotic fragmentation types like ETD or UVPD or IonPD or SID, I don't know if anybody, it seems unlikely anybody has an SID instrument out there, but these are going to look entirely different, right? So these obviously can't be transferred over. So the answer is you can do this if you take some care to see whether the spectra are coming from a similar instrument configuration. But in the end, the highest fidelity match that you will get will be from the same instrument to the same instrument, basically. Some people also ask about iRT, so here's just an example that this actually does work. So this is from this Interlab study that I mentioned yesterday. So this is the distribution of deltas, basically the deviation from the predicted retention time. So when we project using the iRT onto this run, onto the DIA run, where we expect that retention, where we expect that peptide to appear, this is a histogram of how big that error is, and you can see it's like a couple of minutes mostly. And this is our data from Zurich, and this is where the library was prepared, right? So we analyzed all this Interlab data with a single library, and this is data from two other sites using, so it's their DIA data, but with our library, right? And you get basically a similar distribution of errors in the retention time deviation. Well, this one has a little bump over here, something kind of weird going on. And interestingly, we're using a different column, right? So they're using this chip LC from Exigent. We're using a packed emitter, packed with magic. They're using Chrome XP. So the chromatographic conditions are not identical, but it still seems to be transferable to some degree. So the error gets larger. It's not as good, but it still works to a first approximation. On the other hand, if you start changing other aspects of the chromatography, this error is going to get larger and larger and larger. If you start to use a column heater, you change the mobile phase modifier, or you use a radically different reverse phase resin, then this error is going to be larger and larger. So again, it's not like a black and white thing. It's not that you can or can't use prior knowledge from elsewhere. It's just a question of how much it's going to look like the chromatography setup in your system. So again, you can do this kind of thing, but do it with care and evaluate what the errors are kind of likely to be. Okay, so the summary in terms of libraries, there's lots of different publicly available resources. I think this is going to grow rapidly, especially now with Custer synthesizing peptides like a madman. But these resources can be used, but with some care. And the question is, should I always go for the biggest available library for a given project? And well, I would say the hint is here, maybe not. And this we're going to talk about in the next section. So it doesn't necessarily benefit you to take the biggest library and try to find everything that you can, because if there are things, if you're looking for peptides that just aren't in your sample, that doesn't benefit you at all, and it's only going to make the analysis less sensitive by causing multiple testing problems, basically increasing the statistical burden. So these choices need to be made somewhat carefully. I think that probably the safest place to start is by generating your own libraries, which is not usually what people want to hear. But I think this is a good place to start to get a feeling for what's going on, and then these can potentially be supplemented with other resources in a selective way, I would say. Okay, so let's close the library part. Any questions on libraries? Yeah.
Generating your own library means the reproducibility of your results across labs. Like if you're comparing a sample against two different libraries, generate it.
So I think it will cause a change in the results in terms of the set of peptides you can detect. I don't think it's going to shouldn't change the reproducibility of internally of your data, because that's a feature of, how the data was collected and the performance of the machine and various things like that. So I think changing the library is going to change the set of things that you can selectively identify, detect, and quantify in your data. So a mismatch there is going to change the sensitivity in some sense, but I don't think it should change a great deal of reproducibility. I mean, unless people have been using different methods to select which set of fragmentines to monitor, so there might be better or worse ways of selecting fragmentines, which fragmentines to monitor, and that might change the reproducibility a little bit, because some fragmentines might have better reproducibility characteristics or have more or less interferences. I guess, though, does that answer it? No.
The bioprocess people recommend the method that is more spectral libraries that you acquire actually different samples.
Yes.
One that you acquire before some
So they kind of changed their tune a little bit. So they started out by saying, I don't know when you heard that advice, but they started out by saying it was good enough just to do DDA analysis of the samples of interest. And then later on they changed to say, oh, actually, now if we do a little bit of fractionation and analyze that by DDA, then actually there is some kind of benefit. And now in their latest iteration of their software, they have something which is in spirit similar to DIA Umpire, and they now say you can just generate the library directly in the samples. I'm not aware of what they say is better in terms of sensitivity, whether this direct, I think they call it direct DIA or something like that.
Talking about the library from the actual data, but they do a DDA library, and you measure it basically randomized together with your DIA samples.
Okay.
This is more efficient than having a very large library.
Yes.
Yeah,
so I mean, this important question, and it's going to come up a little bit in the next part of the talk. But so the important thing to understand is that there's no reason to have an indefinitely large library that's full of things that aren't in your sample. That can only hurt you, right? So looking for things that aren't there is only going to hurt you. So if your DDA analysis or your DDA analysis of a fractionated sample from your own samples is good enough and big enough to exploit what's available in the DIA, then I agree with that. If your DDA analysis doesn't manage to get all the sensitivity, get all the way to exploit everything that's in the DIA data, then you might see a benefit from going to a larger external library because you'll recover things that weren't in your small library. So at some point there's kind of a crossing over point, and it's actually difficult to know where that will be. And so in our hands, there has in some cases there's been some benefit of going to the big library, but that's only because of probably a deficiency in the sample specific library, if you know what I mean. So where that's going to cross over is actually kind of difficult to predict, and I think this is also something that will come up from Mike as well. But I'm going to talk a little bit more about that in the next section. So now we're going to talk about data analysis concepts, including how to score things, how to do FDR, and how to do sort of post-processing and alignment. So first of all, the rationale, why do we care about it? Sort of objective scoring and FDR in targeted proteomics. Might be obvious, but let's just restate that anyway. We're going to say something about how we extract the data and how we do the scoring. So how do we evaluate what's a good signal and what's a bad signal? And also what you just asked, how do we compute the false discovery rate in targeted proteomics? And we're typically using a Q-value estimation there, using a method called Story-Tibshirani. So did you talk about Q-values and multiple testing correction in the earlier modules at all? Who is in the statistics modules? Did you talk about P-value correction, multiple testing? Okay. All right. Well, some people not, obviously. And then multi-run alignment. So can we borrow power to detect things by looking across different samples? All right, so target analysis concept. So essentially this is what we've been talking about. We have some kind of spectral library, we have DIA data, and we're going to try and selectively extract some set of peptides that we're interested in, and we're going to get some quantitative matrix of confidently detected peptides. So peptide A and B we think are there, peptide C is not detectable, and this is the goal, right? This is the output. And so of course, this, Brendan has now started to show you some data in Skyline, and if you see a signal like this, then you're very happy, life is good. Been not too difficult to see that this is the correct signal without using much external information, but unfortunately it's not really usually so simple as this. So in this case now we have two different peak groups, and they both look pretty good. They're different from each other, they have different ranks of relative ion intensities. How are we going to decide which is the best one? Well, very often what's useful here is referring to this spectral library and saying, do the relative intensities of these fragment ions match what was in the spectral library, for example? And this is what you're going to see in terms of this dot product score in Skyline. So this is kind of the intermediate difficult situation. Well, what about this one? I mean, this is now, is there something, this kind of looks like there's a peak over there, but I mean, is there really a peak or not? So how do we determine non-detectability? How do we draw a line and say something is there or something is not there? And this is what we're talking about essentially in terms of false discovery rate control. So the rationale is also, so Skyline is of course great for visually assessing quality of peptide peak groups, and Brennan emphasized yesterday that it's important to go and look at the data, and I totally will sign up for that. But it is true that it can be subjective, and it's also very time consuming, so this might be possible for SRM data, but when you get into kind of large scale DIA, then you're talking about tens of thousands of peptides, it's sort of no longer possible, and you probably want an objective way to score this anyway. So we need a way to automate the peak detection, we need a way to objectively report the quality of these kind of peak groups, and we need a way to estimate the false discovery rate such that we can make appropriate thresholds, and make appropriate cutoffs and control these types of errors. So what we're going to talk about is automatic peak scoring and FDR assessment. So first of all, let's start with scoring, right? So how do we determine what a good signal looks like and what a bad signal looks like? And so just before we get into that, it's important to, Brendan.
I'm just going to make the distinction that yesterday, when we did start looking at the data, I was looking at only things that the statistical, like I was way down the pipeline where I was looking at only things that the statistical tools had identified as changing, and then I was finding problems with that data. So there's a difference between looking at, you measure tens of thousands of things, you can't possibly look at all of them, but if you don't have tools that allow you to look at your conclusions, then you're still gonna make mistakes. So it's important that hopefully by the time you make a conclusion, you're down to at least a few hundred proteins, and then you could actually look at this. There you want to double
Yeah. Agree. Okay, so first let's talk about decoys. So somebody asked about decoys a minute ago, and the answer is yes, we use decoys too. So let's say that this is our target peptide, AAHTED, whatever, and we know what the coordinates of this peptide are. We've determined this from our spectral library, right? So we're going to monitor this 6 fragment times, and we know what the relative intensities are. We know what the retention time is. But what we're going to do is we're going to create a decoy which kind of looks a little bit like this. In this case, it's using a pseudo-reversal. Actually, we mostly use shuffling of the peptide sequence these days instead of pseudo-reversal, but in this case, it's a reversal that's shown. And what you get is an assay for this peptide, which is kind of somehow related compositionally to this peptide, but it should produce a different signal. And you see what we're going to monitor is the same set of fragment ions from Y7 down to Y3 and B3. But now because we've shuffled around, or we've actually reversed in this case the sequence of the peptide, we have a different set of fragment ion masses. And if you were extracting information at the MS1 level, so here we're showing the precursor mass is the same, but if you were going to extract information from the precursor level as well, you would need to somehow add like some random number or something like this to the precursor mass to make it different from this. But mostly what we're going to be looking at is MS2 level information. So what's important here is that essentially this set of Q3 fragment ion masses has now been altered. We're going to keep the relative fragment ion intensities intact from the target, and we're going to keep the normalized retention time intact from the target. And what we're going to do is going to go to the DIA data, and we're going to extract these fragment ions, and we're going to do it for the target, and we're going to do it for the decoy, and we're not just going to do this for one target, but we're going to do this for all targets, and we're going to do it for an equal number of decoys. So every time we have a target, we're going to create one decoy, and we're going to extract that from the data. And then we're going to rank these. So we're going to look here. So for this given peptide, we have a few different signal groups, different peak groups we call them, which could be the peptide that we're interested in. They have kind of, this one maybe looks a little bit better. This one might be second, and this is third. So we need some way to score these peak groups and say what we think is the best one, which one of these corresponds best to our library, for example. And we're going to do exactly the same procedure for the decoys. And actually, the software isn't going to know what's a target and what's a decoy. It's going to treat everything basically exactly the same way and try to find the best looking peak group for this decoy assay. Try to find the best looking signal. And then what we're going to do is take all the rank number 1 peak groups, and we're going to plot scoring histograms, right? So we're going to have some kind of discriminant score, and I'm going to say where that comes from in a minute now. But we're going to have some kind of score for this particular peak group, which is the target. So that's a red one in this histogram, and we're going to have the same thing for the decoy, and we're going to plot the score distributions of these scores over all targets and all decoys. And if things are working well, then your targets are going to be separated from the decoys. So a good signal is going to the right of this histogram. So this is basically the set of detectable targets, and this little part down here is the set of targets that basically is overlapping with the decoys. So it means that they're not really scoring any better than the decoys. So that means that we don't have evidence to say that we detected those peptides. So this is the basic workflow. A bit more detail about this. So we need these decoys to assess the quality of the peak group scoring and the significance. And the idea here is that these should basically represent. So we use this, as I said, this shuffling of the sequence, or sometimes reversal, but mostly shuffling seems to be the best way to do this. And we generate some kind of non-natural sequence that we don't expect to appear in our sample, but should look like regular target assays to the scoring algorithms. And what that means is that they should have statistically similar properties in terms of how their mass, the Q1 mass is distributed, how the Q3 masses are distributed, and so on. They should basically look like real peptides, but they should not be, we don't expect to find them in the sample. And this is what's depicted here. One thing to point out is that in SRM or PRM, you would actually have to use machine time, right? You would have to use duty cycle to measure these decoys. You would have to set up, configure coordinates and assays and measure these decoys in order to do this. So it's basically double or up to double the amount of instrument time to actually measure these decoys. So that's kind of annoying. In DIA, it's basically unlimited, right? Because all the data is there. You just have to look for these decoys. You just have to say, we're going to try and extract these decoys. And the whole goal here is basically to assess the possibility that we get a signal group of fragment ion coelution that looks like a real signal. What's the rate at which this happens, occurs by chance? Yeah, so this I basically said already. And so now what we're going to do is look a little bit more closely at these scores. And I mentioned this a little bit yesterday, so we have kind of a few different kinds of scores that we can compute. The first category is what we would sometimes call intrinsic, right? So we're just looking at the extracted ion chromatography and saying, is it sort of internally consistent? So do all the fragment ions coelute? And you can see the black ones nicely coelute, but this red one is a little off. Do they have the same kind of shape? And again, the red one is a bit off here. We also sometimes look at the intensity because it tends to be the case that true signals are more intense than false signals. We also have extrinsic scores where we use information from outside, from the spectral library, and I just told you that we were able to know something, we're able to predict the retention time based on previous empirical observations of these peptides using this iRT transformation process. So we can say, does the peptide appear at the expected retention time? So if this is the expected retention time, well, this peak group is closer to the expected retention time than this peak group, so that makes it more likely that this is the correct one. We also, a very strong score, and you'll see this is called the dot product score in Skyline, is do the relative intensities of these fragment ions, so this is kind of plotted in a spectrum here, do these relative intensities of these fragment ions match what's in the library? And this can be a very powerful way to discriminate whether it's correct peptide or not. And also we have sometimes what's called plex scores, so if we spike in isotope label standards, we can directly compare the non-labeled to the labeled standards. And this can be a very powerful way of doing things, but is not employed so often in DIA because of the fact that we're looking at tens of thousands of peptides, so it's just hard to get standards for all these. But it's certainly possible to look at these correlations between labeled and unlabeled peptides. And so basically what we do is that when we have a particular peptide that we're looking for and we extract the ion chromatography, right? So we're looking at these now, in this case, 3 fragment ions, and for each of these candidate peak groups, right? So there's a few different things. Well, it could be this, it could be this, it could be this, or it could be this. So we have 4 candidate peak groups, and we compute all these scores for each of these peak groups, right? For score 1 to 10 or 15 or whatever it is. And basically this is going to help us to discriminate which is the true, using the combination of all this evidence, this is going to help us to discriminate what's the true signal that corresponds to the peptide that we're actually looking for. So we're going to compare, and of course we have the decoys, right? So what we want to do is compare the scores of the targets to the decoys and see if there's some significant increase in the target scores that might be evidence that they're actually present in the sample. And so now I'm going to show a bunch of plots which have this kind of format. So I already showed this scoring distribution. So for any one of these scores that we're calculating, let's take, for example, this dot product score or this relative fragment ion intensity score. You can plot the distribution of those scores for all of the peptides that you're looking for, and you can distinguish these. You can label them as targets and decoys. And as I mentioned, if things generally, so in dot product, a score of 1 is the best, right? So if it's over to the right here, it means it's getting a good score. If it's somewhere over here in the middle, it means it's not really distinguishable from the decoy distribution, so that's not good evidence that the peptide is actually there. The other type of plot we tend to use a lot for this is a ROC plot, and this is basically a plot in some known kind of like gold standard data where we know what's true and what's false. We can plot the false positives versus the true positives, and so essentially what we're doing is walking from the from the right to the left down this score and saying, are we getting true hits or false hits? And if everything is working well, then this line is going to go up the y-axis like this, and if it was perfect, it would go basically all the way up the y-axis and go across like that. If it's random and we don't have any information, it's going to be on this diagonal. So typically what we see is a line something like this that indicates that we have some information. So the larger the area under this curve, under this green line, the better performance of this particular score in separating the true from false targets. So for any of these scores, we can now plot one of these little histograms. So these are all these scores that I mentioned, and we can now transform these into these histograms and these AUC plots, right? So for each of these different scores, these different characteristics, we plot targets and decoys, and we figure out which of these scores are the most informative in terms of recovering the things that we know are true positives. And we can also overlay all these plots. So now this is the overlay of all these scores, and these are all the different scores that I talked about. And you can see that the worst performing ones are going to be down here. The best performing ones are going to be up, kind of hugging this y-axis, going up into the corner here. And so, yeah, you can see, for example, like I mentioned, this intensity correlation. So this dot product score is a very powerful one. This, if you had, in this case, this is a reference shape score. So I think this is talking about a isotope label reference, but it doesn't matter. And then finally, what you would actually like to be able to do is combine all of these subscores into a single score. We need a single discriminant score. Yeah. Yeah. Yeah, so, well, so the ideal thing there is to not change the fragmentation pattern, right? So the ideal thing will be to use the same collision energy, and then you would have good fidelity between the spectrum library and between the ranking and the DIA data. But if you are using spectral libraries from another source, or for whatever reason the collision energy is different, then you won't have a perfectly good match between those. However, it still might be better than no information at all. And this is basically what we're assessing here. So in your case, let's imagine that we made this plot for a library where the collision energy was different, or it was from a different instrument. What you would see is that this yellow intensity correlation score would probably drop down here and would be a less good performing score in that case. However, so what we're going to do is combine all of these scores into a single discriminant score in a way that maintains the most information. So if the information is there, then the algorithm is smart enough to use that information in a weighted way. So if that's a very informative piece of information, then the algorithm will rely on that more highly. If it's less informative, then it will assign it a lower weight, and it won't use that information as heavily in this final discriminant score. Brendan.
But also, if you built your library from DDA on your same instrument, then all these instruments have a function for figuring out what this collision energy should be based on the precursor energy. And then what they do in DIA mode is they're going to use there are two mechanisms. They're either going to use the CD for the central thing, and then actually the one difference is that you probably have to choose a charge state to optimize for. So we generally choose charge state 2, and yeah, that may not be exactly the same CE that was used or would be used in DDA, but it will be the central charge 2 collision energy for the range, and then also I think SciEx does a ramp. So they'll ramp over the whole range. But these are pretty small ranges, and the CEs are going to be pretty close to what you would get in DDA. But it's true that, like, the optimal CE for a charge 3 might need to be on a completely different equation, and it might be, like, a little lower than charge 2 or something like that. And so, yeah.
So one trick we sometimes play to get around that is we set up the DDA charge state calculations to treat everything as if it's +2. So this is actually suboptimal in terms of identifying higher charge state peptides, because the higher charge state peptides will be over-fragmented. But for the ones that you do identify, the fragmentation pattern will match very well the DIA data, where you can't make these decisions that are based on charge state. You just have to treat everything the same. So that's one thing you can potentially do.
I think some of these things are covered in the paper.
Yeah. And also, one last thing to say, actually, and I don't know whether Mike will show this data, but this is different. This is mostly where we're talking about beam type CID, like HCD, or QTOF type fragmentation. But some people have also started to use ion trap, like on a Lumos instrument. You have the option to do fusion or Lumos instruments. You have the option to do the fragmentation in the ion trap using CID, using resonant ion trap style CID. And there you don't really have this issue so much because everything is fragmented in a resonant way, so it's hard to over-fragment peptides. And so I think Mike has some data to show that higher charge state peptides might be performing a little better in this type of fragmentation. So that's an interesting kind of direction to go. I don't know if, did we actually answer your question, or do you want to? Yeah, well, so that's something that we do in our lab. I don't know if we ever really systematically evaluated whether that's the best thing to do or not. But I think it's important to understand, and I'm going to go on to this in a minute now, that there are other scores than just this library relative fragment time intensity correlation. And even if you remove that score from the algorithm completely, it would still actually perform pretty well. The sensitivity might go down a little bit, but there's still many other characteristics of the data that you can score. So it's not just this, right? So even if this is not perfect, the algorithm will try and compensate for this by not using that information, not relying as much on that information. Okay, and so this is kind of what I'm getting to now, is that so we have like these 10 or maybe 15 different scores depending on which software you're using, and we need to combine these into a single score, right? So in order to be able to plot these score histograms here, we need to use a single score. And the question is, how are we going to combine all these like 10 or 15 different scores into a single discriminant score? And this kind of depicted pictorial here, that these, if we plot score 1 versus score 2, they have different kinds of information, and that the combination of those two scores can give you a better discrimination than either one of them would do alone. So what we'd like to achieve is the combination of these two scores in this plane here, except now it's like a 10-dimensional space here, or even a 15-dimensional space, but we want to basically find a combination of these scores that best discriminates the targets and the true targets and the decoys. And what we use for this is called linear discriminant analysis. So this is essentially a machine learning technique where we label part of the data set as true, and in this case we're talking about some subset of the targets that we basically are pretty sure are correct. So we score them with a pre-score, or even with one of these pre-scores, the most powerful one. We take the highest scoring set of those and we say, okay, we're gonna accept that these are true, and then we take some part of the decoys and we say these are false. And now we're going to try and optimize the combination of all these subscores in a way that they best separate these two labeled sets of data. So this is semi-supervised learning. And what this looks like mathematically in the end is you have score 1, 2, 3 to 10 or 15, and what we're trying to learn is a set of coefficients, right? A set of weights that when we sum up all these scores together, so this is a linear combination of these scores, we get a discriminant score that best separates the true and false. And so this is what I mean, that these kind of like whatever scores are better or worse internally in your own data set, the algorithm is smart enough to try and figure out what are the most informative scores in that case. And finally, we now take this final discriminant score, which we call sometimes the D-score, and we make these plots of targets and decoys. And now what we need to do is use these histograms to estimate the false discovery rate. So, one thing to mention is, like, how do you know if these decoys are fair? I think, and this is an interesting thing to occasionally look at, and the way we usually assess this is we try to take a library from like a completely different species, and we use that library. So in this case, we're talking about DIA data that was collected from human samples, and we do a lot of work from M. tuberculosis in our lab, so we have this TB library lying around, and we try and extract a set of targets from this TB library. And what you can see is that, so this score distribution is basically the same as this histogram here, but now shown as a density plot. And so the yellow is the human targets. So here we see a kind of bimodal distribution where we have a yellow part which looks basically like the decoys, but we also have a yellow part which is shifted to the right, and this is the set of things that are actually detectable, the set of human peptides that are actually detectable. And blue is the human decoys, right? So they basically follow this yellow part here, the non-detectable targets. But now we have the MTB targets and decoys, and they basically both look indistinguishable from the human decoys. So that's a good sign that the decoys that we're using for the human library are reasonably fair, because if we take a set of real target peptides, naturally occurring target peptides that we expect to occur somewhere, but not in our sample, and we try to extract them, well, then they should basically look the same as decoys. So this is kind of a good reality check sometimes, I think. Okay, so most of the scores I mentioned are the kind of chromatographic scores, but there are also, which you could have calculated like on SRM data, but this is just for completeness to say there are some other sets, some other scores that we can only calculate when we have high resolution MS2. So the mass accuracy, Brendan talked a little bit about yesterday, the isotope distribution. So does the isotope distribution match what we would expect for a fragment ion of that size? And in addition to these 6 fragment ions that we're going to extract, well, we would expect to see the whole rest of the B and Y ion series, or some part of the B and Y ion series, in the MS2 spectrum, so we can go back into the high resolution scan and check are there any more of these expected B and Y ion fragments there. Also, these days, we sometimes extract information from the MS1. Sometimes we do, sometimes we don't. It's particularly useful in some settings, like, for example, in the PTM world, it can be very helpful to have these precursor scores. So we can score things like, do the precursors coalute with the fragment ions? We can also score like mass accuracy and isotope distributions from the precursors. And so this is kind of an optional extra in open SWATH, which we sometimes switch on or not, depending on the data set. Okay, what about computing FDR? Okay, so we're not doing too bad. So computing false discovery rate and targeted proteomics. So we're going to use, as I mentioned, the Story-Tib-Shirani method for estimating Q values. So what's the definition of FDR? So this is essentially the fraction of reported peptides in the final output that are wrongly assigned as detected in the sample. And in FDR estimation, so somebody asked in DDA, right, you can use mixture models or decoy counting to estimate this. In targeted proteomics, decoy counting without any kind of adjustment, which I'll mention what this means in a minute, will be rather conservative, and I'll try to explain why that would be. Okay, so here's the procedure for estimating FDR. So here's one of these target and decoy distributions, these histograms. So gray is targets, red is decoys. So you can plot these histograms, and we recommend that you do look at these histograms. You'll see these in Skyline also later. So take a look at these, and you can plot these, and you can see, I don't know how easy it is to see, but basically you can see that for the targets, for the gray, this is essentially a mixture of 2 distributions, which is the true targets represented by this population shifted to the right here. So remember, going to the right is increasing discriminant score, so better evidence that the peptide is detected. And we have a second population here, this purple in the purple outline, which has essentially the same distribution shape as the decoys. So this is the set of things that we don't have good evidence for saying that they're detected. And so essentially what we do is we do use the decoy distribution to compute p-values. So the decoy distribution is used as an empirical null, and we calculate p-values. So for any given peptide, every peptide is going to have a specific d-score, right? It's going to fall somewhere on this x-axis, and we're going to compute p-values under this empirical null distribution for whatever that score is, right? So if you have a score over here, you're going to get a very small p-value under this empirical under this null, and if it's over here, you're going to have a non-significant p-value. And then we use a method called Story-Tib-Shirani to transform these p-values to q-values. So this is a multiple testing correction, and this depends in a large extent on what we call the fraction of false targets. So this purple, this false part of the target distribution, if you compare that to the decoy distribution, this false part of the target distribution has a smaller magnitude, and the difference in magnitude between this part of the target distribution and the decoys, so we refer to as the fraction of false targets, right? So this part of the distribution is the targets that we can't, that we don't have evidence for detecting, and the question is what fraction is of that, of all of the targets, or in effect also of the decoys. And effectively, you can imagine what we're doing is scaling this decoy distribution to essentially match these false targets. Conceptually, this is essentially what's going on. So what's the Q value? Well, the Q value is a p-value that's been corrected for multiple testing. So functionally, the Q value has this very convenient property that if you filter your data at a given Q value threshold, the FDR, the false discovery rate, in the remaining data is equal to whatever that threshold was. So if we filter all of the peptides at a Q value of 0.05, then the false discovery rate in the remaining data, after we've thrown everything away that has more than 0.05, should be 5%. And same deal for Q value 0.01, then the remaining data should be 1%. So this is a very convenient property of Q values, and it makes it easy to filter the data. And so the way that this actually works is that this story-tip-Shirani method, what we're doing is making histograms of p-values. So we take all of the p-values that we computed, and we make a histogram of this. And what this histogram would look like if there were no true positives in the data, right? So let's imagine none of our targets were detectable in the data. What we would have is a uniformly distributed set of p-values. So what we'd expect is just to get p-values across the whole range from 0 to 1. And the issue is that, let's say we made our significance 0.05, we would still have a bunch of stuff which is below 0.05, even though there isn't really any true positives in this data. And so this is what the score distribution would look something like for this equivalent to this p-value histogram, right? So here we have the targets in gray and the decoys in red, and these score distributions are essentially indistinguishable from each other. So we don't have any gray targets over to the right here, and this is basically why we have a set of p-values that is basically uniformly distributed. So we don't have any evidence for detecting anything. However, hopefully your data will look something a bit more like this. So if we plot the p-value distribution for a real data set where we have targets that we expect to detect, well then the density of this p-value histogram shifts to the left, and we have a lot of things that are actually significant. And the fraction of false targets we estimate, so this is the level, if all of the assays, if all of the peptides will be false, then this is the level we would expect to see this histogram at. And we now estimate the proportion of false targets, right? So by looking basically at where this histogram flattens out, this allows us to make an estimate of how many false targets there are there. So how many things are there that we're looking for that aren't actually detectable in the data. And we use this correction factor to correct for multiple testing. So what about context? So in the past, like when these methods, at least in our lab, were developed mostly for looking at SRM data, where we didn't have that many targets, and usually most of the targets were detectable, and so this worked pretty well. So we didn't have a lot of problems just estimating these errors at this kind of so-called peak group or peptide query level. And what we usually were doing, we're also looking at the FDR in kind of one sample at a time. So we didn't necessarily have really huge studies where we would propagate these errors as we built up a lot of samples. But when we went now into DIA mode, where we have lots of peptides, we have large libraries, we're looking for things that aren't there, we have heterogeneous data, these false discovery rate estimation methods we discovered were not really sufficient. And this is kind of something that Rudy alluded to yesterday. So the question is, does this actually make a difference? So changing the nature of the data, scaling it up, and having lower prevalence data, like having libraries where we're trying to extract targets that aren't actually in the sample, does that make any difference in the false discovery rate? And the answer is yes, but well, it depends on a few things. The size and the comprehensiveness of the spectral library, and this comes back to the point I made earlier, that you don't necessarily want to have an indefinitely large spectral library. You don't want to be looking for things that aren't in the sample, because this is only going to increase the statistical burden. The number of samples and the heterogeneity of the composition of those samples has an impact on this, and the relationship between, as I said, the peptides that are present in the samples and that are represented in the spectral library all has an effect on inflating FDR estimation under these kind of assumptions up here. So okay, what do I mean by that? So in the case where now we have DIA data and we're going to build a sample-specific library, and we can use that sample-specific library to do kind of a targeted DIA data analysis, and it's likely that the set of things in the library should match pretty well the set of things in the DIA data. Well, and basically the samples in the study are pretty homogeneous. That doesn't really cause much of a problem. What becomes a problem is essentially when you have a library. In the first instance, if you have very heterogeneous samples, right, these are compositionally different, then you're going to get a kind of more larger and diverse. So it's still going to be a study-specific or a sample-specific library, but it's going to be larger and diverse because of the compositional diversity in the samples that you're studying. And so this is kind of less targeted in a way with respect to what's in the library compared to what's actually detectable in the DIA, right? So the library is going to be a bit bigger than what's detectable in any given sample. And then if you go really large scale, like these were kind of repository scale spectral libraries, then you're going to have a very large library, a very heterogeneous kind of library, and you're going to be extracting from a set of samples which are kind of homogeneous and have, so the prevalence of things that are detectable in these samples is going to be much lower than what's actually in this large scale spectral library. And so this is what's kind of depicted here again in these density plots. So these are now density plots from DIA data where we extracted the data, and here we're using a library that was generated directly on the same samples. And so what you see is the targets are basically all shifted over here to the right. So we can basically detect almost all of the targets, or like 95% of the targets. We have good evidence that they're all present. And we use this pi0 annotation to denote the fraction of false targets, right? So you have like 5% of things that are sitting underneath the decoys here and essentially not detectable. Go now to now a really large scale spectral library. So let's say this like combined human assay library, and then we try to use that to extract. Well, now this distribution looks very different. So this is the set of detectable things in the library here, but what we mostly have is targets that we don't have evidence for their presence in the sample, and so we say that the pi0 is large here. So like 60% of the things in our library, we don't have evidence that those are detectable in the sample. And the difference between these has kind of implications that you might expect from the way this false discovery rate is being calculated, right? That the value of this null part has an effect on the multiple testing corrections. So the fraction of false targets, so the larger this pi0, the higher the statistical burden in terms of multiple hypothesis testing. And this is what I'm saying, that the more things that you look for that aren't there, the larger the correction that you're going to have to make for multiple testing. What about from sort of scaling up from like peptide query to protein FDR? So if we plot the false discovery rate at the peptide query level, or sort of the peak group level, up to versus the protein false discovery rate, you can see, and we know why this is, right, from what I said earlier and from what Rudy said. So if we take a 1% peptide query FDR, and we look over here, we're getting a 5% protein level FDR, and we know why that is from earlier in terms of the sort of non-uniform mapping of true hits and false hits from peptide to protein level, but that's for one run. So now what happens, and basically this is, I basically already described this earlier, so I'm going to skip over that, but if we now want to do protein level FDR estimation, the method that we do to do this is just to take the best scoring peptide and replot these histograms essentially. So we collapse all of these peptide queries down to the protein level by just taking the best scoring peptide hit. So this is a very kind of simplified way, but it's actually a reasonably effective way to do this. And now we get another scoring distribution like this, and we can repeat this Story-Tip-Shirani Q-value method, and we can estimate Q values at the protein level. And so what I'm trying to get at now is that basically when you go from one run to lots of runs, and if we're calculating these peptide and protein FDRs on a run-by-run basis, then we're going to build up as we add more runs, we're going to build up protein false positive disproportionately as we add runs, right? So now for a 1% peptide query FDR, now we're up to above 20% protein FDR. So this relationship between peptide query and protein level FDR scales badly as you add more runs. So what's the solution to this? So this is basically what Rudy showed in this slide yesterday, that we discovered this when we started to move to larger data sets. Well, first of all, what's the reason for this? So here's kind of a toy example where we have 5 different samples, and again, what we have is all of these true positive hits. We tend to see the same ones over and over again, right? So if this is each row represents one protein, and a green is a true positive in any of these samples, and we again have this problem that the false positives tend to appear in different proteins when you go across different runs, but the true positives always appear basically in the same proteins. So when you actually go to the kind of global context, when you count, so if you look at like the matrix, and you say how many false positives are there in terms of rows of the matrix, well, now your FDR is going to be significantly inflated. So here in this case, on the sample by sample basis, we had 20% protein level FDR, but when we go to this kind of global context, this is now, by aggregating all of these false positives together, you now have a 42% protein level FDR. So the question is what we can do about this, and the answer is to kind of calculate protein false discovery rate in different contexts. So this is a little bit complicated. I'm going to forget where the references is down here. I would recommend, if you're interested, take a look at this paper. This is also the one Rudy cited yesterday, which goes into a lot of detail of this. But essentially what we realize is that we can calculate false discovery rates in different contexts. So we can ask the question, is a protein detected in a specific sample, meaning that we just look within this one sample, and with this score histogram and so on that I've been showing, we calculate that only from the targets and decoys within this one sample. We call this run-specific context, and the question there is a protein detected in a specific sample? Or we can go to what we call this experiment-wide context, where we consider basically all of the runs, and these score distributions that I showed are built by aggregating all samples all together, and the question here is, in which subset of samples is this protein detected? And this can be useful, but in order to really control this, when you go to large-scale data, you need to go to this global context. And the question here is a protein detected in at least one of the samples? And so the way that this works is we collapse all of these samples down to the best scoring hit for a given peptide or protein across all these samples. We just take the best scoring hit, and now we build these scoring and FDR, these scoring histograms, and the FDR estimation is done only on the best scoring hits per sample at the peptide and protein level. And this is what we call the global context, and this is very helpful for controlling across the whole study the false discovery rate. So now I'm going to go back to this. Okay, I'm running out a little bit out of time here, but I'm now going to go back to this interlab study that I described yesterday to illustrate this. And so now I told you about this already, but from the perspective of this, what I want to show here, basically we're only going to be looking at this complex HEC293 background, and we're going to say that we have 229 measurements of this, essentially replicate measurements from different places of this HEC293 lysate. And we looked at this. So this goes a bit to the question of what kind of sizes of libraries you can potentially use. And we looked at this data with three different libraries. So the largest one was this combined human assay library, which contains 10,000 proteins. The smallest one was a sample-specific library that we generated from DDA analysis of just a few, like 5 injections of this HEC293 lysate. And then we made a kind of intermediate-sized library, which was a subset of the yellow one, but we just took out all the DDA runs that have been generated using HEC samples. So this is kind of a HEC-specific subset of the larger library. And we did the analysis, and essentially this is what the scoring distributions look like, at the protein level for these different libraries. So this is just to illustrate to you the effect of this different pi zero, the magnitude of the fraction of false targets, essentially, the fraction of undetectable targets. And so for this sample-specific library, and so the decoys basically look all roughly the same, right? But the targets for this sample-specific library, you can see that most of the density is over here on the right, so we're basically able to detect everything in the sample-specific library, almost everything. For this hex subset, we have kind of a somewhat of a bimodal distribution. That's this purple one. So we can detect most things from the hex subset library. But from this large combined human assay library, we can only detect about half of the proteins, right? So there's 10,000 proteins in this library, and we can detect about 5,000 of them. So this essentially makes sense. And the point here is that each of these different libraries have a different pi0 associated with them, right? So this fraction of targets that we cannot detect is quite large for this large combined assay library. It's a bit smaller for this HEC293 library, and it's basically pretty close to 0 for this sample-specific library. And I don't know if Rudy showed this slide or something like it yesterday, but basically what we do now is we compute the protein FDR using this. We have 3 different ways to control the FDR, right? So this is if we just look at the peptide query level within, we're computing this for all files, all 229 files individually, or if we add in protein level FDR control, but again, only on a file-by-file basis, or if we add in this global protein context and we say we're just going to constrain the analysis to the set of things we detect, the set of proteins we detect at 1% global protein FDR, which is about 5,000 proteins. And here you can see these saturation curves that we showed yesterday. So essentially the behavior that we would expect to see, because this is basically replicate analysis of the same sample, what we see is that this one which is controlled in this global context, saturates very quickly. And this is using this large library, I should say, this combined human assay library. This saturates very, very quickly, but with these other methods, which are less strict, we basically get the number of proteins detected going up into the sky indefinitely, and eventually we'll probably get up to all the proteins in the assay library. But this is not, this is an obvious sign that we have a lot of false positive rates. So introducing this global protein FDR when using this large-scale library has been very important. We can also overlay the plots for these other libraries. So for the smallest library, all the lines are sitting on top of each other, right? It doesn't really matter. You can use any of these methods to control the FDR, and they basically all look the same, and that's because of this small pi0, right? Everything that's in the library is basically detectable. And the intermediate library has some kind of intermediate behavior, but the difference between the kind of global FDR control and the other two is a bit smaller than it is for the largest library. So this is just to emphasize to you why it might not be useful to necessarily use the largest library. So we do see some sensitivity benefit. So notice, right, that there's a big sensitivity benefit. Maybe it's clearer. No. Notice that there's a big sensitivity benefit to using these larger libraries sometimes, but we do have to take care in terms of controlling the FDR. And if this local kind of sample-specific library was better, then that would be optimal, right? So what you want in your library is the set of things that are detectable in the DIA data and not more. And of course, it's hard to know what that is, but that's what you would be aiming for. That would be the most efficient thing in statistical terms. So this is this paper. So I mean, what I explained there is now pretty complicated, but if you want to understand that in a bit more detail, I would recommend you take a look at this. The high-level message is the FDR can be inflated if you analyze a lot of samples, especially heterogeneous samples. Then you want to do global FDR control. And if you're using a large library where you have peptide queries, you're looking for stuff that isn't there in the sample, then it's important to calculate protein level FDR. And I would say it doesn't hurt to calculate this all the time, actually, even if you don't have those conditions, because it actually doesn't cost you that much to calculate these things. Okay, last thing. So I'm 5 minutes over, but I'm just going to spend 5 minutes on alignment, because it's also worth talking about, and then it's going to be coffee. All right. So multi-run alignment. So the goal basically here is to increase data completeness and consistency. So even though we don't have this stochastic, DDA problem that causes missing values in DDA data, sometimes we still do sometimes have missing values because we have to make thresholds. We have to say there's a false discovery rate threshold, and sometimes a peptide will be above or below that, and then you get a missing value. So what we can try to do is look at the experiment in a more integrated way and say if a peptide is detected in this sample, well, maybe it's more likely to be detected in this the next sample as well. So we borrow some confidence essentially from one file to the next one to try to change the thresholds at which we say something is detected or not. And that's the purpose of this trick algorithm, which is called transfer of identification confidence. And so basically what we're saying is that if this peptide is detected confidently here with a very good Q value, and in the next one, for whatever reason, the Q value is above the threshold that we set, well, if it's at the same retention time essentially and looks the same, well then it's likely to be the right signal, so maybe we can recover this and we can extend our threshold and say we're going to keep this one because it looks very much like a confidence signal in the next run. What this requires in practice is very good chromatographic alignment, because we want to, with high fidelity, be able to say that this is the same peak group as this one, even if there's a chromatography shift. And so I talked to you earlier about iRT, where we have linear transformations of the chromatography space, but to get really good accuracy in terms of these transformations, we now go to nonlinear alignments at this stage. And it's also possible, and this is kind of an optional step, is if there's a sample where there's basically no signal, we don't see anything, it's possible to say, based on the other runs, well, this signal should be here in the retention time space, so we could just integrate whatever value is here and use it as a noise value to complete the matrix and to do the statistics. This is a little bit risky because you can, if this nonlinear alignment is not working perfectly well, then you could end up over here and get all sorts of weird interfering signals in there. So this needs to be used with a little bit of caution. So the way this works is basically what we first do is take all the runs and we compute an optimal guidance tree. So basically we do a run-to-run pairwise distance matrix in chromatographic terms. So which runs look like each other chromatographically, and that's using all of the detected peptides now. So it's not just using these 11 iRT peptides, but it's using all the peptides that we can detect in all the runs. And what it does is look for the common set of peptides, high confidence peptides, which are overlapping, and says which runs are kind of more like each other in the chromatographic space. And we build a tree which basically says a minimum spanning tree, where each node is one run, and the connection is basically the shortest chromatographic distance between these different runs. And so this is just to guide the alignment. And what happens is that for all edges on this minimum spanning tree, we compute nonlinear alignments, right? So we have all these potentially thousands of peptides identified in all runs, and we can make plots of run 1 versus run 2, and we can fit a curve like a low S curve or a spline or something like this, and we can do transformations very accurately between run 1 and run 2. And so we do this for every peptide, and we use this guidance tree as a way to guide this. So we take basically the run that has for a given peptide, we take the run that has the best score for that peptide, and we propagate that confidence throughout the rest of the runs in the experiment according to this tree, right? So we have a nonlinear alignment from run 1 to run 2. We have another alignment from run 2 to run 3, and so on. And we're basically able to propagate the confidence there. And this allows us to achieve this effectively. So this is the confidently identified peak group. This is the less confidently identified peak group. But because it's chromatographically in the same space, and the error is low because of this more accurate nonlinear alignment, we can sort of extend the threshold for this peptide and say, okay, this probably is the same one as this one, and we're going to include it in our quantitative matrix. And for a run where we don't have a signal, we can also integrate that. And and so this is just a kind of a demonstration of this, that if we use these kind of hard thresholds, where, for example, we were to say, oh, we're going to throw out everything that has a Q value which is larger than 0.01. This is now like 180 yeast runs. This is the number of peptides detectable, and you can see it kind of goes up and down a bit. But after we apply this alignment, we're able to rescue some of this data, and we now have a much higher completeness in the quantitative matrix, right? So this is the matrix completeness, goes from 0.59, so 59% to 83% at the peptide level, and from sort of 80% to 92% at the protein level. So this is a useful thing especially for large data sets, to kind of make our quantitative matrices more interpretable and more amenable to statistical analysis. Okay, so I'm going to just summarize now. So what are we doing? We're doing automated chromatogram extraction. We're scoring the peak groups from targets and decoys in the spectral library. We're doing the semi-supervised learning step to calculate the best discriminant score that we can based on the separation of targets and decoys. So what's the best combination of all these subscores that's going to give us the best sensitivity? We use FDR estimation. We estimate Q values here using this Story and Tibshirani approach by first computing p-values and then correcting those p-values for multiple testing. The context of the FDR matters, how you calculate this. Especially, it makes a big difference if you have samples where you're looking for a lot of things in the library that you can't actually detect. So we call this high pi zero, right? So there's a large fraction, there's a large null fraction. So our recommendations would be to control the FDR on a protein level and do it also globally to prevent error accumulation when you add new runs to the data set, essentially. And the quantitative data consistency and completeness can be improved by doing this transfer of identification confidence and more accurate across run retention time alignment. Okay, so that's it. I'm sorry I went a little bit over time there. Are there any burning questions, or do people want coffee and to turn their brain off for 10 minutes? Yeah, okay, let's do that. Thanks.